Hello, those of you who have joined. I'll wait a minute for more people. So far we have 10. But now it is time to start. First, I'd like to make sure you all remember that today is quiz 6. You have all day to take it. And I have assigned another worksheet. This one is on genetics, problem solving, and genetics. So you'll find that in Canvas. And after today's class, you'll have a much better idea for how to do the genetics problems. And speaking of problems, I'd like to come back to the membranes. Most of you turned in a worksheet on membranes. Are you hearing me, by the way? Anybody not hearing me?
I can hear you.
Okay. And so what I thought I would do first of all today is go over the problem solving with regard to membranes, because there does seem to be a lot of confusion about how to do those problems. And I will have one or more problems like that on upcoming tests. So first I want to share my screen. And now you should all see worksheet diffusion. Anybody not see that? Okay. So you all did well on the multiple choice down to the problems. So the first thing I want to point out is this is taken from one of the slides from the lecture, Guide to Osmosis and Tonicity. So everything you need to know to solve these problems is here on these statements. First, osmosis only refers to the water concentration. And tonicity only refers to the solute. And the third point is that substances always move down their concentration gradient. That is, from high concentration to low. And the final point is that a solution has two components, the solute and the solvent. So if you know the concentration of one, you immediately know the concentration of the other. So for example, if the solute concentration is 60%, then you immediately know that the solvent concentration is 40%, because the total always equals 100. So let's go through. There are three problems here. The first one, the dialysis bag filled with 0.2% glucose placed in a beaker containing 10% glucose. Now, the glucose cannot go through the bag. So I've drawn a little picture here. I have found that it's really helpful to draw a picture of the problem so that you can visualize what's going on. So the bag has 2% solute. And so that tells you that it's 99.8% solvent or water. So and the beaker is 10% solute. So that means that it's 90% solvent water. And the glucose can't move through the bag, but the water can. So since the concentration of water in the bag is higher than it is in the beaker, the water is going to move out of the bag. It will go down its concentration gradient from the bag to the beaker. As for tonicity, tonicity refers only to the solute. And so if you compare the two solute concentrations, 0.2% and 10%, 0.2 is low compared to 10, so the bag is hypotonic, and that makes the beaker hypertonic by comparison. So that's how you should think about this kind of a problem. Anybody not understand how to do this? Any questions?
This is Steve Stinson.
Uh-huh.
I don't really get it. Sorry.
Well, okay. Which part of it, can you identify which part you don't get?
I guess I don't get the way this solute is supposed to move.
Okay, in some cases, the solute will move. In this case, it doesn't. So this is a dialysis bag, and it has a pore size that allows water to go through it, but the glucose can't go through it. So solute does not move in this example. We have another case where solute does move. But in this case, do you see that? You see why solute doesn't move? It's because of the nature of the dialysis bag.
Okay, that makes sense.
Okay, so then the question is, which way does water move? And so you have to think about the fact that, go up here to this little table, that a solution has two components, the solute and the solvent. So if you know the concentration of one, then you immediately know the concentration of the other. So in this case, the solute concentration in the bag is 0.2%. So that means the water concentration there is 99.8%. You see that?
Yes.
Okay, water concentration in the bag is 99.8%. In the beaker, it's 90%. So you're comparing 99 to 90, and which way is water going to move? It's going to move down the concentration gradient, which means it'll move from 99 to 90. And so the question is, which way does water move? The answer is it will move out of the bag. So does that make sense?
Okay.
Yes, it does. Thank you, Professor.
Okay, so then with regard to the tonicity in this case, hypo means low, hyper means high, and this refers only to solute. So where is the solute low? You compare 0.2% to 10%. 0.2 is lower than 10, so the bag is hypotonic, and the beaker is hypertonic. Does that make sense?
Yes.
Okay.
No.
No? Okay, what doesn't make sense? Can you identify what doesn't make sense? Okay, well, think about that while we go on, and we can come back to it. Let's go to the next example. In this case, we have two beakers connected by a tube, and in the tube, there is a membrane. And in one of the beakers, there's 10% fructose, and in the other beaker, there's 0.5% fructose. Now, both fructose and water can go through this membrane. So in this question, it's as though the membrane isn't even there, because both water, both solvent and solute can move freely between the beakers. So the first question is, which way does water move? Well, if it's 10% fructose, then it's 90% water. And on the other beaker, if it's 0.5% sucrose, then it's 99.5% water. And water is going to move down the concentration gradient. So the concentration gradient in the right beaker is 99, the left beaker, it's 90. It's going to move from 99 to 90. That means from right to left. Which way will fructose move? Well, it's going to, again, move down its concentration gradient from 10% to 0.5%. It will move from left to right. And with regard to tonicity, it refers to only to the solute. So 10% is higher than 0.5%. So the left beaker would be hypertonic. Hyper means high. And the right beaker would be hypotonic with respect to fructose. So does that make sense to everybody? I want to make sure everybody understands how you think about these problems.
Okay, you saying that both the water and the sugar is going in the beakers at the same time at the bottom here, right? And then 10% of it, which is on the left, is going to the right, correct? And 5%
Yeah.
is going to the left because hypo one hyper. Which
Yeah.
Is,
Well,
okay.
but hyper and hypo are not related to the direction of movement. So for the direction of movement of fructose, it's going to move down its concentration gradient, which the gradient is from high to low. So it's going to move from left to right.
Because it's more, is that right?
Yeah, it's more higher concentration of fructose in the left beaker. So it's going to move toward the lower concentration, which is in the right beaker.
I guess.
Yeah, this example was a bit challenging for me. I had to personally draw it out and try to like visualize it.
Yeah, who's speaking?
This was Nicholas Gonzalez.
Nicholas Gonzalez.
And then the first person was me, Yolanda.
Yeah, I got you.
Okay.
So Nicholas, you
Is that
say, go ahead.
Is that the way you wanted us to explain it on the worksheet?
People
Like with
explained
the
it
drawing?
in various different ways. Nobody drew pictures. That would have made it easier for me, and I think you too. But what I did was read your answer and tried to interpret it with respect to the questions, and I gave you credit where I thought you had it right. So anybody that thinks I didn't grade you correctly, let me know.
So it's okay if we draw pictures too.
Yeah, that would really be helpful in most cases, especially in cases like this. But we're going to have more problem solving when we come to genetics. And again, drawing a picture often
no.
helps.
Thank you, Professor.
Okay, let's go through the last question. That's about the sheep red blood cells put in a beaker containing a solution of 0.2% sodium chloride. So again, I drew a picture of it here. Here's a beaker of 0.2% sodium chloride. And the red circles are the red blood cells. And so we have to think about what are the concentrations. The question wants to know which way is water going to move, and then what are the tonicities? Well, we know that blood cells are about 0.9% salts. Any cell is about 0.9% salts. And put in a beaker with 0.2% salts. So the solute concentration in the beaker is lower than in the cells. And conversely, the water concentration in the beaker is higher than the water concentration in the cells. So which way is water going to move? It's going to move from the beaker into the cells, and that will cause them to swell. And probably with these concentrations, they'd probably burst. Then if we talk about tonicity, refers only to solute. So which tonicity, which solute is in a higher concentration? 0.9% is higher than 0.2%. So the cells are hypertonic, hyper meaning high, and the beaker is hypotonic. So everybody see that clearly?
No. You say it's already got water in the container, but you said because the solute hyper is 9%, those little red things. Now they're already underwater. They're gonna swell because of the 2% hypo.
Leave hypo out of it when you're talking about which way water is going to move. Only think about the concentration of water.
Professor,
Yeah.
so everything, like anything always goes towards where there's less of that thing, right?
Yes.
Okay, that makes more sense. So that's why the water is going in the cells, because the cells were used to less concentrated water.
Yeah. There's
Okay.
less water in the cells than there is in the beaker. And so water is going to move, again, think down a concentration gradient. So the water is going to move into the cells.
Professor, this is Alicia Carroll.
Uh-huh.
So that's how the cells would be hypertonic because the water would be moving into it, even though water only refers to osmosis. We're still talking about the water moving. Is just the transfer of water.
Yeah, always separate the movement of a substance from the tonicity. Don't think about them together. So if you want to think about which way is water going to move, think about the water concentration in the beaker and the water concentration in the cells. So the water concentration in the beaker is 99.8% water. Whereas in the cells, it's 99.1%. And so water is going to move from its higher concentration to lower. The higher concentration of water is in the beaker. The lower concentration of water is in the cells. Water will move from the beaker into the cells. And so think about the movement of substances separately. That keeps it simple. And then you come to the next question about tonicity. And that's always you're comparing concentration of one with concentration of the other. And the higher concentration is always hyper. The lower concentration is always hypo.
This will be on the test.
There will be questions like this on the test, not on the quiz today, but on the test coming up, test 2, there will be at least one question like this, maybe more.
Monday, right?
No, that test 2 is October 27th.
Oh, 27. Okay.
Excuse me,
Hopefully
I
go
want
over
to
the
say
game.
something.
Uh-huh.
You know, like you can see like the more like explanation in the book and tonicity and the osmosis, you know, it's really like explained and really like illustrated.
The explanation in the book is
Yeah,
easier to understand.
yeah, I did like more and I see like, you know, like at the tonicity, it says like the animal cell can like burst or rise when it's like when water comes to it, like
Mhm. Do you have the page numbers in the book that you could tell the rest of the class?
yeah, it's a chapter 3, and I see that osmosis is page 79. Yeah, and all these, you know, are explaining almost all the worksheet that you gave us.
Okay, well, thanks for pointing that out.
No.
And so for the rest of the class, if you're still having trouble, refer to the book. You can also Google this. Just ask for either tonicity or membrane transport, and you'll find several other ways people explain these how to do these problems. So if you're still having difficulty after my explanation, there are other explanations that you might find more helpful.
Okay, so the answer you were looking for, what will happen to the cell is they are going to swell. Is that what you were looking for?
Yeah,
The
but
answer?
do you know why they're going to swell?
Okay, and that was because of the water and the
Sir.
fluid.
Uh-huh.
Professor, is any possibility that before the test on class, can you explain some of those questions? So make another examples of these.
You want more examples of tonicity and
Yeah.
substance movement?
Yeah, it could be before the test because you know, because this is only a few questions, and in the test, when we have the test, it's really hard, and especially the time behind us. And for me, it will be better if you make another example in next class, if you can.
Okay, I can do that. The question on the test will never be the same as an example that I discussed. So of these 3 questions,
That's
none
why
of
we
them
get
will be
confused.
exactly like this on the test, but it'll be, what I really want you to know is what it says in this guide. And if you understand these points, then solving these problems is always the same. You go through the same steps. If you separate movement of substances as one part of the question and tonicity as the other part of the question, you'll get it right every time. But yes, I'll
Okay.
make a note to do up another example at the beginning of class next time.
Thank you.
Okay. So I want to move on now. Today's a short class, 60 minutes, because you have a quiz. Do you see Mendelian genetics? Tell me if you don't.
No,
You don't see it?
no, I don't see anything.
Okay, then I'm going to
No.
stop.
I saw it.
Let me try again. Do you see it now?
Yes, I can see it.
Okay, anybody that cannot see a slide that says Mendelian genetics at the top?
No, I could see it.
Okay, everybody can see it. All right.
Yes.
So I want to talk about Gregor Mendel because he's where Mendelian genetics comes from. But I also want to mention him within the context of evolution, Charles Darwin. And we'll talk a lot more about him later. And I just want to make a point about history here, that these people, these two men worked in the mid 1800s. They didn't know each other, but this was an era of great ideas. And I just mentioned a couple of others, great ideas and great events. So for example, Karl Marx in this same period was thinking about economics and writing about it. And he posed a number of theories about that, which are still affecting policies and politics today. And of course, in this country, we had the Civil War in the middle of the 1800s, and that profoundly affected our history. And the effects of that are still going on today. So the middle of the 1800s was very eventful. Getting back to Mendel, he published his work between 1856 and 1863. And he had no knowledge of DNA or of genes or chromosomes, but he used a little plant, garden peas, in a very clever way in order to discover the evidence for genes. Now at the bottom here, Mendel's laws, so we'll talk about his laws later, but even though they were published, nobody paid any attention to his work for about 40 years. And then about 1900, 3 different scientists from different countries repeated what he claimed in his work and found that his results were entirely repeatable. So suddenly Mendel's laws, Mendel's observations had credibility. And scientists, that's when genetics really took off. Now, Charles Darwin, as I said, he didn't know Mendel, but he was the driving force behind the theory of evolution at the same time, and the concept of natural selection. So the results of these two men have established the basis for biology ever since. So at the bottom here in yellow, Darwin established natural selection as the force that controls evolution, controls species, and Mendel provided the rules, the evidence that there are discrete entities that determine species. Okay, prior to Mendel, there were plenty of people interested in heredity. How do progeny come to be the way they are with respect to their parents. And so what people who are interested in this would do is take two parents with different traits and cross them. Could be plants, could be animals, but cross them and then look at the progeny. And what they would always see is continuous variation, no segregation into distinct groups that could be related back to the parents. So I've drawn or shown you here a picture of a bell-shaped curve where parent 1 was crossed with parent 2. And if you look in the progeny, you see that on the one side of the curve, the progeny are like parent 1. And at the extreme other end, they're like parent 2. But in between, it's a mixture of parental types. So instead of getting discrete phenotypes, what you get is a smear of traits going from like one parent all the way to like the other parent. And nobody could make any sense of this. This was just the way biology worked until Mendel. And he did two very insightful things. They're listed here on the left. He did inbreeding and he did analysis of the second generation, which is F2. But first we have to think about some definitions. What is a cross? It's a breeding of two plants or animals, one with another. The parental generation is the progeny of a cross, and it's called an F1, filial generation 1. If you take two progeny, two F1s, and cross them with each other, F1 times F1, you get an F2. So the F2 generation is the offspring from crossing two F1s. Phenotype and genotype. Phenotype is what you see. What you see is a pink flower or a purple flower. The genotype is a symbol. Genotype is a symbol which represents the actual genetic makeup. And genes are represented by letters. So you see that here. The phenotype can be tall, short, black, pink is what you see. The genotype is a letter. So in the case of a tall plant, if tall is dominant, you give it a capital T. And if short, then it would be recessive, and that would be a lowercase t. But genotype is a letter. Now, how did Mendel do his experiments? It's important you understand how he did it to know how he got his results. He first developed what are known as true breeding parental strains, also known as inbreds. So on the left here, what is an inbred? It's the mating of two closely related individuals. So that after several generations of inbreeding, all genetic heterozygosity is gone, all the loci are homozygous. The result is a true breeding line. In contrast to that, a hybrid is the mating of distantly related individuals, and that results in most loci being heterozygous. Now let's take a look at a pea. Peas are self-pollinating. That means that both the male and the female organs are in the same flower. So the ovary is there, the stamens are there that make the pollen. And so before the flower even opens, all the fertilization is done. All the eggs get fertilized by the pollen. So this was a problem that that Mendel had to deal with. So I've talked about several terms here. I'd like to explain them. Zygosity and alleles. So homozygous means two identical alleles of a particular gene. So the alleles are identical if it's homozygous. Heterozygous is the opposite. It means that two different alleles of a particular gene. And alleles are simply alternate forms of a gene. So does everybody understand these terms? Terminology is really important in genetics. And I have a table of terms at the end of this slide set for you to consider, think about. Okay. This illustrates what alleles are. So we have two chromosomes here in purple, and these chromosomes are homologous. That is, all, what is homologous? The alleles are identical.
Yeah,
And
they're the
so-
same. Okay.
Yeah, yeah. So these chromosomes are homologous, which means all of the genes on both of these chromosomes are identical, except for one, the allele for purple flowers and the allele for white flowers. So those two alleles are different, so they are considered heterozygous alleles. Remember, heterozygotes are different forms of the same gene.
I have a quick question.
Yeah,
Can alleles be mutated?
yeah.
Oh, okay. I also expected a much longer answer, but never mind. Thank you.
Yeah, well, it's a good question, because any gene, since it's made of DNA, any gene can be mutated. And so if you have, so for example, supposing in this illustration, both these alleles at this locus, this gene, had been for white flowers. Well, one mutation in the white flower allele could lead to purple flowers, and that would make it heterozygous at that locus. Okay, let's go on to inbreeding, because this was key to Mendel's success. If he hadn't done inbreeding, well, nobody prior to him had done it. And if he hadn't done it, he wouldn't have come to his results. So without this, his distinct segregation patterns would never have been seen. So I've already gone over this. What is inbreeding? It's crossing closely related individuals. An inbred is an individual that results from repeated inbreeding or backcrossing. So if you keep mating an individual to itself for many generations, for example, 6 generations, now all the loci are homozygous on all chromosomes. So what you want to have before you start doing crosses, if you're Mendel, is homozygous parents that are homozygous at all loci. So they're pure, they're genetically pure, and they're known as true breeding. But what are the characteristics?
Is that the same for animals?
Yes, same thing can be done with animals. I mean, it's not as
okay.
easy, but the principle is the same. You can take a mouse, for example, and cross a mouse with another mouse, take the children, cross them to each other, keep crossing, and keep crossing, generation after generation, and you end up with mice that are homozygous. All their genes are identical on both chromosomes.
How many times do you have to inbreed something for it to become Homozygous.
The generally accepted number is 6.
Okay. All right, that's what I,
But there could be more or less. The more you inbreed, the more completely homozygous the individual is. But if you do the math, by the time you get to 6 generations, if you start with 2 parents that are completely heterozygous, by the time you get to 6 generations, they're 99.9999% homozygous. Okay, look at these pictures. These are the traits that Mendel started with. So he looked at seed shape, and some were round, and some plants had round seeds, some had wrinkled seeds. And when so first he inbred. So he had seeds that were 100% homologous for round and differed only by 1 allele for wrinkled. And when he crossed them, all the progeny were round. And similar with seed color, yellow times green, all the progeny were yellow. And then flower color, purple and white. The F1s were all purple. Pod shape, pod color, flower position, and plant height. Again, for each of these, he started out by inbreeding, so that he had pure lines of round, pure lines of wrinkled, pure lines of tall, pure lines of short, and everything in between. So he didn't start making crosses until after he had done all of this inbreeding. And then when he did his crosses, since this is a self-pollinating plant, he had this very tedious procedure he had to do. So before the flower opened, the pollination occurs before the flower even opens. So before that happened, he would have to take forceps, open the flower with his hands, take forceps and remove the stamens. The stamens are the ones that make pollen, the male cells. And then if he wanted to do a cross, he would take the pollen from one flower and open the flower of a different plant and sprinkle the pollen around on the other plant. And then the seeds would develop, he'd get seeds in a pod. So this process involved opening the flower buds, removing the stamens, dusting the pollen from one plant, the stigma of another. This procedure was extremely tedious. Imagine doing it with just one flower. Mendel did thousands of these, many thousands. So let's look at the result of his first experiment. It's called a monohybrid cross. So this is key step number 2, F2 analysis. Remember, key step number 1 was inbreeding. So he's done that, key step number 1. And now he's going to do key step number 2. So he took a plant that was now homozygous for purple flowers and crossed it with one that was homozygous for white flowers. That means they're highly inbred. And we looked at the progeny, they're all purple. Now remember, in the past, when people have made crosses without inbreeding, they would have seen everything from purple to white and various shades of color in between. Mendel saw just one class. Every progeny of this cross was purple. And then he did something that, again, no one had ever done before him. Instead of stopping at this point, he said, what happened to white? So he did another generation. He took purple progeny and crossed with other purple progeny. And now amazingly, white appears. This is the F2 generation. And he counted. And when he did, he always found a 3-to-1 ratio. There would always be 3 of one color to 1 of the other color.
So the purple was the dominant one, in other words.
Yes, purple is dominant.
Okay.
So for the first time, progeny appeared as distinct classes rather than as a continuous variation. Now that shows the phenotypes. So let's take a look at the genotypes. And in this case, we're going to call the purple plants big P for purple, big P because it's dominant, and we're going to call the white ones little p because they're recessive. So if you cross big P, and we know it's big P because it's inbred. We know it's little p because it's inbred. And so the purple parent can only make big P gametes, sperm and eggs. The white plant can only make little p. So if you cross a big P and a little p, there's only one possibility, and that is a big P, little p progeny. And that is the F1. So the F1 is heterozygous, it's all purple, but the gametes that the F1 can make, half will be big P and half will be little p. So now if you cross two F1s, you're crossing a big P and a little p, and you get the following result in the F2. So if you look here at the eggs and at the sperm of the pea plant, you're crossing big P, little p with big P, little p. So if a big P fertilizes a big P, you get big P, and that's purple. If a big P fertilizes a little p, you get big P, little p, and that's purple. And the reciprocal is also true. But if a little p fertilizes a little p, you now get the genotype little p, and that's white. Everybody see that? How we get 3-to-1 ratio. The phenotypic ratio is 3 purple to 1 white. The genotypic ratio is 1 big P, 2 heterozygotes, and 1 homozygous recessive. This is the only one that's white.
And this is the monohybrid cross.
Yes.
That's what this is called. Okay.
Yeah, monohybrid means that only one gene is involved. All the rest are homozygous. So how do we define dominant and recessive? The way Mendel did it, he said that the characteristic that appears as the phenotype in the F1 generation is dominant. The characteristic that disappeared in the F1 and then reappeared in the F2, he called recessive. And those are still the terms we use today. Well, here, this illustrates dominance and recessive on homologous chromosomes. So the pink one and the blue chromosomes are homologous at all loci except the ones indicated here. So on homologous chromosomes, any given locus can be either homozygous or heterozygous. So if you look at the P locus, it's homozygous dominant. So it's big P on both chromosomes. At the A locus, it's homozygous recessive. It's little a, on both chromosomes. And at the B locus, it's heterozygous. It's big B on one chromosome and little b on the other. So this illustrates how on a set of homologous chromosomes, you can have both homozygous and heterozygous loci. Okay, genotype versus phenotype. On the left are genotypes, on the right are phenotypes. So look at the phenotypes first. There are, in the progeny of the F2, there are 3 purples and 1 white. And the reason for that is because of the genotypes. The genotypes are a homozygous dominant, heterozygous, 2 heterozygous, those are all purple, and 1 homozygous recessive, that's the only one that comes out white. So let's look at an example. We have a true breeding, means an inbred, tall P, crossed with a true breeding short P. Tall is dominant to short. What will be the genotypes and the phenotypes of the F1 offspring and the F2s? So you need to work through this yourselves, but I show you here how to do it. So you know that this true breeding tall plant, it makes gametes, makes eggs and sperm. And because it's dominant and it's tall, you give the letter T, big T. And the plant itself, because it's diploid, is big T, big T. And correspondingly, the true breeding short plant is little t, little t. They're both a diploid, so they both have 2 copies of the gene. Now to get the F1, you cross a big T with a little t, and the only genotype possible is the heterozygote, big T, little t. And the phenotype would be tall. But now, if you do the F2, you're crossing big T and little t. You get the gametes of the F1 are both big T and little t. And so there are 4 possibilities. And the best way to sort these out is to draw a picture in the form of what's called a Punnett square, where you have parent 1 on one axis and parent 2 on the other. Now in this case, both parents are heterozygous. This is for the F2, remember. So you're crossing a big T little t times a big T little t. And so if a big T sperm fertilizes a big T egg, you get the homozygous dominant, big T, big T. If a little t parent, a little t sperm fertilizes a big T egg, you get big T, little t. You can get the reciprocal of that, big T, little t. And only if a little t sperm and a little t egg fertilize, you get the homozygous recessive. So this is how you get the 3-to-1 ratio in a monohybrid cross. And the worksheet that you have as your assignment has a whole series, I think 9 different questions, different genetics problems that you should work through, and we'll go through them, I think, in the next lecture period, so that you can all see how to work through crosses. And let's see, we're coming close to the end of our time. But you need to know what is a test cross and why you would do a test cross. How can we determine an unknown genotype? So the example here is you have a plant with a purple flower. Now you know that that plant must be either homozygous dominant or a heterozygote, but you don't know which. So you cross it to a white plant. And why white? Because you know the genotype of white has to be homozygous recessive, little p, little p. Now, if this purple plant is homozygous dominant, big P, then you're crossing a big P with a little p, and all the progeny will be heterozygotes. That means they're all purple. And so if you make this cross, all the progeny are purple, then you know this parent was homozygous dominant. If, on the other hand, it's a heterozygote, then when you cross big P, little p, with little p, this is the white one, you're going to get half and half. You're going to get half heterozygotes, which will be purple, and half homozygous recessive, which will be white. So that, if you get half purple and half white, you know that this plant was a heterozygote. So that's a test cross. And some of the problems you will have on your sheet, and which might come up on tests, involve the use of a test cross.
Now we would have to do that diagram, or would we just have to put little p is going to be the white?
It depends on how the question is asked. In some cases, I might ask you to do the Punnett square, and in other cases, maybe not. But in any case, you should, because that's how you come up with the right answer.
Okay.
So what happens if you're trying to follow more than one characteristic at the same time? This is an example where one set of characteristics is smooth seed coats with a yellow color, and the other plant has wrinkled seed coats and green. And what you end up with is a 9:3 to 3:1 ratio of smooth yellow, wrinkled yellow, smooth green, and wrinkled green. And this shows how you get there. And again, you end up with this Punnett square, which helps you to visualize. First of all, it helps you to get all the genotypes right, and you can interpret from the genotypes what the phenotypes would be. And so you can see that you get 9 smooth yellows. You can count them, 1, 2, 3, 4, 5, 6, 7, 8, 9, and 3 smooth greens. Here's 1, 2, 3, 3 wrinkled yellow, here 1, 2, 3, and only 1 wrinkled green. And you get that by plotting the genotype of one parent against the genotype of the other parent, and then just working out one at a time what the genotypes are. And from the genotype, you immediately know the phenotype. So there will probably be a dihybrid cross somewhere in your future.
I have a quick question. This is Anthony Imelo.
Uh-huh.
So when you get 9 smooth yellow phenotypes, is that essentially telling you that that is the main phenotype of these, or?
You have to compare the all the phenotypes with each other.
Okay.
So just looking at one class doesn't tell you much. You have to look at all of them and count. This was one of the things that Mendel did. He counted each category, and that's how he came up with these ratios. And then because he got these different ratios, he worked backward and came to the concept of a gene. He didn't know he was doing that, but he came up with the concept that there are discrete entities controlling traits. So there were just discrete things within the sex cells that were controlling what phenotype you see in the progeny. So these are the laws, Mendel's laws of inheritance. You should know these. They are segregation, independent assortment, and dominance. These are the three conclusions that Mendel drew. So what is segregation? Allele pairs separate during gamete formation. And remember, we've just spent a whole section talking about how gametes form in meiosis. And these gametes unite at fertilization. And so these are two sources of genetic variation. One is recombination in tetrads during meiosis 1. And the second is fertilization, because any egg is fertilized by a random choice of sperm. I've taken pictures from the previous slide set to illustrate this. Between anaphase and telophase, chromosomes segregate. And after recombination, the segregation results in variation. Similarly, another of Mendel's laws is independent assortment. Independent, emphasis. Chromosomes separate randomly from one another at metaphase. So this is another source of genetic variation. You see that in the lower right, when chromosomes line up in metaphase in the center of the cell, any chromosome can go either one way or the other. So this red chromosome could go to the top or it could go to the bottom. And that is entirely random. Similarly, this recombinant chromosome, a blue-red chromosome, it can go either to the top or to the bottom. So this is another source of genetic variation, and because of it's entirely random, it's called independent. And the law of dominance, we've gone over that. The dominant allele masks the recessive one, and the recessive one appears in the F2. There's one other thing. I'm not going to be able to finish this in this period because our time is up. But I just want to point out that even though I've emphasized dominance as an absolute characteristic, it can have variation. So dominance is one allele masks the other. Codominance is when both parental phenotypes end up in the F1. Incomplete dominance is when you actually get a blend of both parental phenotypes. So this can be confusing, but you can figure it out. And then there is sex-linked inheritance, and we'll talk a little bit about that next time. But that's all I have for now. I'm going to stop there and remind you to take quiz 6. And we'll talk again on Monday.
Quick question.
Professor?
Yeah.
Hey, you got it, man.
No, go ahead.
I was just gonna ask, are we gonna practice doing Punnett squares?
Yes.
Okay. So is it in like the next worksheet?
Yeah, the next worksheet that I think it becomes available today. And yeah, work through those as best you can on your own, following the rules that we've gone over today. But then we will discuss them in class.
Okay.
And there will be questions like this on the next test.
All right.
Okay, Professor, I have questions at the end of class.
Okay. All right.
Have a nice day, Professor.
Okay, I'm going to